Chapter 16, Part 4, T lymphocytes. These are also just called T cells. These are made in the red bone marrow. They mature in the thymus. That's where they're called the T cells. They circulate in the lymph and in the blood. They often migrate into lymph nodes or the spleen. They'll hang out in Peyer's patches, which are little lymphatic areas in the mesentery of the intestines. These guys all have a protein called T cell receptors on their cytoplasmic membranes, and those T cell receptors are going to interact with some of those MHC proteins. And the T cell receptors are pretty specific. They don't recognize epitopes directly. They don't just specifically bind to an epitope floating around in the plasma, for example. They have to bind to an MHC protein first, and then they can check its epitope. So the T cells get triggered and act against cells that have intracellular pathogens or that are producing abnormal cell surface proteins, things that things they shouldn't make. Viral infected cells are making the wrong things, and these T cells and the T cell receptors detect what they're making and say, that's not supposed to be there. Now this is what the T cell receptor looks like. It's embedded in the membrane at one end. These are again separate proteins all held together by disulfide bonds. We have constant regions, which are constant in this person. All of their T cell receptors are going to have the same exact constant region, and that's generally the same for all people. For humans, there's a constant region. It's the same for all people. And then there's variable regions, and these are variable not just between different people, but every single T cell has a different variable region that it produces. That's how variable it is. Every single T cell generates a new variable region. And that means that every one of them recognizes a different antigen, has a different antigen binding site. They recognize different antigens. These have carbohydrates on them. That's what the little green balls are. But this is going to very specifically recognize one antigen that fits in this antigen binding site. Now, functionally, there are different types of T cells. They're functionally different, and they have some different glycoproteins on the surface of them, which is how we identify them. So we have, again, these are all T cells. We have cytotoxic T cells. These directly kill other cells. So these cause the immediate death of cells right next to them, and then they get out of the way so the cleanup crew could come in. Helper T cells. Boy, these guys are really important. They coordinate the whole system of T cells, all the types of T cells, B cells, macrophages. They release cytokines that recruit other cells from the immune system. The helper T lymphocytes are really important for coordinating a response. And as we are seeing, if you have a coordinated response against an infection, and this includes a pandemic, if you have a coordinated response and a coherent one that is effective, it will work, and you will reduce that infection, and in this case, you will eliminate a threat. And so with the helper T cells are super important for coordinating that response. And when there are no helper T cells, then the response is not coordinated, and then people get sick. And I say that because in HIV, these are the cells that HIV knocks out and kills. These are the cells that they infect. HIV infects helper T cells, and then they die. And that reduces the whole immune system's ability to respond to any kind of pathogen. The pieces still work, but since they're not working in coordination, they don't work very well. And that's when you get infection. And that is the characteristic of AIDS, right? It's having this virus HIV, but then also getting sick with things that normally don't make people who are healthy sick. So that's my little side note there on the helper T lymphocytes. So there are even different types of helper T cells, but they are all involved in coordinating the whole immune system. There are also regulatory T lymphocytes, and these ones are important because basically the cytotoxic T lymphocytes get kind of excited and they go out and go on a death rampage, and that's okay when there's an infection. That's okay, but then at some point it's all over and we need to calm everybody down, and not just these guys, but all of the other, the neutrophils that are all ready to go and attack, and all of the other white blood cells, they need to be repressed a little bit periodically to just go back to normal. This fight's over. Nothing to see here, folks. Move along, move along. That's what the regulatory T lymphocytes are doing. And this is a little table of the characteristics of the types of T lymphocytes. These guys are all T cells, so they all mature in the thymus. They have different kinds of surface glycoprotein, CD4 or CD8, and then what other things they also secrete. So when T cells are being developed, that variable region of their T cell receptor, that's randomly generated, completely random. There's a whole genetic manipulation thing that happens at the DNA and the chromosomes where the genes for the variable region are. They get cut and pasted in a bunch of ways as that cell develops into a T cell, and they develop independently but completely random T cell receptor variable regions. So every single T lymphocyte is unique in what its antigen binding site is. And that means that a lot of your T cells that you make just randomly will recognize normal cell proteins, normal things that are supposed to be there. And if they get, what the T cell's job is to find things that it recognizes and then kill the cell that has it. So it's really important that you kill off the T cells that make antigen binding sites on their T cell receptors that are going to be recognizing normal cellular proteins. So early in the process of becoming a T cell, there's a screening process where basically all the T cells say, here's my epitope, or here's my antigen binding site. And if this is normal, then you need to kill me, and that's exactly what happens. The ones that have antigen binding sites that bind to epitopes that are normally produced in that individual's body, those T cells die. This is called clonal deletion. So, because if you don't do that, now you have a T cell who's going to go out and find that normal thing that on a cell, and then it's going to kill that cell. And then it's going to find the next cell that makes that, and it's going to go kill that cell too. And once they get to a certain point of development and they can't go through clonal deletion anymore, if we missed it, now we have autoimmune disease. Now we have a T cell that is recognizing and targeting for death normal cells that make a normal protein in your body. So that's something we really don't want to do. So the body eliminates self-reactive lymphocytes. So all the lymphocytes that react to autoantigens undergo apoptosis. That's programmed cell death or cell suicide. So these cells, most of them, don't make it through this process because it's random, and we have a lot of really normal stuff that these guys can, by random chance, make antigen binding sites that they recognize them. By the way, this clonal deletion, we're going to talk about B cells soon. Clonal deletion happens the same kind of way with B cells. Now they're going to have a different way of responding and a different antigen binding site, but they have an antigen binding site too on a B cell receptor. And those B cells that detect antigens that are are clonally deleted during development as well. So this is something that happens in both T cells and B cells. And it's really important that it does, because again, if you don't, now you have an autoimmune disease. So there are 4 possible fates for T cells. The cells that don't recognize the MHC protein, they can't recognize that. Well, they're not going to be able to do this job anyway. So they go through apoptosis. They die. The T cells that recognize autoantigen, normal stuff, they also die by apoptosis. Some of the self-recognizing T cells, most of the T cells die, but some of those, they become regulatory T cells. And then some of the T cells that aren't any of those, they recognize the MHC protein, so that's good. And they recognize foreign, not self epitopes. They become your protective T cells that circulate in your body, move into your lymph nodes, hang out, screen the fluid that moves through the lymph nodes on its way back to the circulatory system. Okay, so here's what happens as the T cells become T cells. We have a stem cell and it produces T cells. Each of these T cells, again, is going to have a T cell receptor with a different binding site on the T cell receptor. And first they're challenged, do they recognize MHC? And if they do not recognize the MHC, then there's no reason for them to continue, and they are caused to go into apoptosis. If they do recognize the MHC, then they get checked for, they survive that process. And then they get challenged, do you recognize autoantigens? And if they do, then most of them are caused to go into apoptosis. A few of them become regulatory T cells. If they do not recognize MHC autoantigens, then they are allowed to continue to grow and to become mature, active T cells. And the next section, we're going to talk about B lymphocytes. Just to be clear, the T cells are involved in cell-mediated autoimmunity. The B cells are involved in humoral immunity. So the T cells find out what the cells are making with this MHC proteins, and then do whatever needs to be done to that cell or to the neighboring cells based on what they find. The B cells are going to sit and hide in places, in pockets in the lymph nodes and the spleen and wherever they want to be, but they're going to generate cells that migrate out and that produce massive numbers of antibodies to target and opsonize an invading pathogen.